gang, how's it going? We're 17 days into self-isolation. Hopefully you've been entertained. You're doing a little bit of studying maybe since you have a test coming up. But I'm just going to go through Unit 2 today. I'm not editing this video like I would normally. So if I make mistakes along the way, just roll with the punches. So Unit 2 is all about cell structure and function, and that is almost self-explanatory, right? So know about the parts of the cell and what they do and why they're important. So I'm going to go through this. We're going to talk about different cellular components and their functions, cell interactions with their environment, cell membrane structure function, regulatory mechanisms like osmosis, selective permeability, and compartmentalization. Now, I'm not reteaching, I'm just giving you guidance as to what you should review. Whoa, see? Okay, so when looking at the cell structures, we can limit our list to these right here, right? So ribosomes, which we know make proteins, endoplasmic reticulum, which we know compartmentalizes the cell as well as transports those proteins, the Golgi apparatus, which packages and manipulates the protein, so maybe it adds a carbohydrate to it to make it a glycoprotein. Mitochondria, which we know make energy, lysosomes, which have digestive enzymes, and so they break down molecules, chloroplasts, which are for harnessing the sun's energy and making carbohydrates, and then vacuoles, which are storage containers. And then you could talk about the central vacuole of the plant cell, which expands and maintains the turgidity of the cell. You could talk about contractile vacuoles, like in a paramecium, that expel excess water. So each of these things, you want to know what they do, maybe be able to describe their structure and identify why they are able to do what they do. Describe the structural features of a cell that allow organisms to capture, store, and use energy. So right away, you're going to think of chloroplasts and mitochondria when you read that, right? So one of the big things in this is surface area, and how the surface area, by increasing surface area, you're increasing the area that can do work, right? So both of these, this is the thylakoid. Each one of these is a thylakoid all put into a granum, the sac. And then you have like the double membrane system of the mitochondria and the folded up cristae. So you want to think about how this folded business is increasing the surface area. So there's more ATP that can be made here. There's more of sun's energy, and ATP can be made. So I think a lot of the AP likes to focus on hierarchy of organization. So anytime you can identify pieces that add up to bigger pieces, just like cells to tissues to organ systems. Like you have a chlorophyll, which goes into a photosystem, and the photosystem goes into a thylakoid, and the thylakoid goes into a granum. Anytime you can see those layers, you want to be able to discuss those layers. Oh, I'm not very good at rolling. So surface area to volume ratio, remember the larger surface area to volume ratio is better, and you might want to be able to calculate that, although if we're limiting what we're going to ask of you come April this year, 2020, I don't know that you'll be calculating surface area, but some things that add to surface area, one is just the shape of a cell. So like the red blood cell has that concaved business, so that's creating a foldedness. Your brain, so this is where you can connect to some of those body systems, and like somebody asked how the body systems fit into all of this, because they're not like identified as a big topic, but they're examples to be used within other topics. So like surface area is one of those examples. The folding of the brain increases surface area. This fold, there you have inward folds of your stomach, as well as you have microvilli, like finger-like projections sticking off of your small intestines, which allow for more digestion to occur. There's cilia, there's root hairs, all of those. I have root hair picture on another page, but you know, you have your main tuber root maybe, and then little hairs going off of that, so they increase surface area, more chance to absorb nutrients from their environment as well as water. Increasing your cell size decreases the surface area, so cells are limited to their size because they become less efficient. Specialized structures, so this is where we might talk about root hairs. So here you can see extensions like these cells. Notice how that cell has an extension, so that's increasing its surface area. Lots of osmosis could occur with the environment. Ions can be transferred through those roots. So it's kind of like spreading itself out like a web to capture as much as it can. Some specialized features of plant cells or leaves are those guard cells right here, right? We know they're part of the stomata. They create the stomata that opens up and lets gases in. And so the guard cell can fill its vacuole and open, or it can release its vacuoles and close. So that's going to create an efficient exchange of molecules with the environment being oxygen and CO2. The fluid mosaic model, so you want to talk about the pieces and parts that are in the fluid mosaic model. The phospholipid, which is its basic structure, the embedded proteins, which serve as enzymes or markers or identifiers or transporting material through the cell. You have glycoproteins. Glyco is usually related to communication, so glycoproteins are glycolipids. That would be a glycolipid there, a glycoprotein here. Kind of like antennas, help for cell communication. Cholesterol, which is embedded within the lipids, and that maintains the fluidity of the cell membrane. Also remember, like your hydrophilic portions and your hydrophobic portions. So hydrophilic will orientate itself towards the exterior of the membrane or the cytoplasm inside. The hydrophilic will orientate itself to the inside of the membrane. And that's true of the phospholipids as well as the proteins. So these portions here that are on the surface are hydrophilic, and these portions in the middle are hydrophobic. Explain how the structure of biological membranes influence selective permeability. So you're looking at polarity, right, of molecules and of membranes. So the small nonpolar molecules, like oxygen gas, for example, they pass freely across the membrane. I have seen this question in a FRQ before. Hydrophilic substances or large molecules, so if they're polar or large hydrophilic, they need an ion channel to pass through. If they're too large to fit through the pores, they need a transport protein simply because they're too large. If they're charged ions, they have to go through a channel because they would be repelled by the hydrophobic intersection, right? So polar molecules need channels for that purpose. The structure of the cell wall, you can think not all cell walls are made of cellulose, right? We talked about plants having a cellulose cell wall. Chitin is found in fungi, and peptidoglycan is found in bacteria. And here's two different bacteria, the gram-positive and the gram-negative. So the purple is the peptidoglycan. So here you see peptidoglycan on the surface, and it's in layers. And a lot of times the structural components are in layers, right? We talked about how cellulose forms layers. So that's all talking about how it's structured. Here, the gram-negative, that peptidoglycan is on the inner surface. So we know membranes form permeability. Cell walls form more structure, as well as a permeability barrier. Mechanisms that organize to maintain solute and water balance. So when you read this, automatically you're thinking of osmosis and diffusion. So we know they're ruled by concentration gradients. Here we have a concentration that's high in solutes. Here it's low in solutes. So if things move from a high to a low concentration, that's passive transport. It does not require energy. If it's moving from a low to a high concentration, you're shoving more particles into a crowded area already. That's going to require energy, and that'll be active transport. And we know that a cell uses ATP for energy. If it's a large molecule or a polar molecule, it uses a facilitated diffusion. It uses a carrier protein or a transport protein. It's still moving it from high to low, so it does not require energy. If this was a pump, the word pump is usually going to indicate an active transport going in the other direction. Endocytosis and exocytosis, when the membrane itself surrounds particles exterior and brings them in, that's endocytosis. And exocytosis, a vacuole makes its way to the surface, binds to the cell membrane, and then releases its components on the outside, and that would be exocytosis. That does require energy, though it's not considered active transport. Explain how the structure of a molecule affects its ability to pass. So this is where if it's positive or negative, or if it's neutral. So we know if it's polar, you have to use facilitated diffusion. Water molecules, they'll pass through a little bit all by themselves, but if you need a large amount of water to pass through, you might use some aquaporins. Those are transport proteins that fit just right with the water molecule. So the movement of ions also affects the polarity of the membrane. So the polarity of the membrane referring to the charge. So this is again where that nervous system becomes an example. So for that, we have typically more sodium on the outside of the cell than inside. So the outside of the cell is positive compared to the inside. When sodium rushes in, then the inside becomes more positive than the outside. So you reverse the polarity. That's called depolarization. And then to reset that, you have to kick the sodium back out, and that's going to, I got to include potassium here. So when you, for the depolarization, sodium comes in. Repolarization, potassium goes out. So another positive has left. And then that creates a hyperpolarization because usually there's an overshoot, and then the sodium-potassium pump reverses it and resets the cell's membrane potential or its membrane polarity. So if that sounds totally unfamiliar to you, go back and watch the video on action potentials. Okay, how concentration gradients affect the movement of molecules. So this is referring to tonicity. Remember, tonicity is referring to how much solutes there are. So referring to the environment, hypertonic, there's lots of solutes. Hypo, less solutes. Iso, they're the same. So we know if you are in a let's see, this one is hypertonic. The water will leave the cell becomes crenated, and the cell shrivels. Or in a plant cell, it becomes plasmolyzed. This one is in a hypotonic solution. So lots of water rushed in, the cell exploded, or cytolysis, or here it increased its turgidity. And then if it's isotonic, they're moving in and out equally, and so it doesn't change its shape. So you should know the rules and be able to talk about the rules. You don't just take for granted that the reader understands the information. You have to tell the reader what you know. You're giving points for correct information. So when talking about the movement of molecules, talk about concentration gradients. When talking about water, talk about water potential. So we know water moves from a high water potential to a low water potential. Another way of saying that exact same thing is to say water moves from low osmolarity to high osmolarity. So osmolarity is referring to the solute concentration, right? So if it has low solute, it has lots of water. Okay, so be able to talk about those two concepts. Practice when I'm done talking today. Osmoregulation maintains water balance and allows organisms to control their internal environment. Here's a couple examples. Again, this is a piece that we didn't get to yet, the body systems. So this is where the body systems become an example of the other topics. So when you are maybe dehydrated, for example, your kidneys will try to conserve water. And so they hold on to that water by releasing less in the form of urine. Another thing that's happening is you have like ADH, antidiuretic hormone. So antidiuretic hormone is released from the brain and travels to the kidneys, and then that opens up those aquaporins, so more water is reabsorbed. Aldosterone is another example of a hormone that's being released, and that causes vasoconstriction of vessels, and so less water is lost in that sense. Animals that live in freshwater, for example, would definitely need a way of regulating their osmoregularity because of diffusion of solutes or the intake of water. So if water and solutes can move into their skin, they need to be able to get rid of it, right? So inside of their skin, like inside the cell, or the body of the organism, you have way more solutes than you do outside. So water is, of course, going to come in. So they need a way to expel it. So that also has to do with osmoregulation, maintaining water pressure, basically. Comparing a few things here. Describe similarities and or differences. So comparison, contrast. If FRQ asks you to compare two things, identify the similarities. If it asks you to contrast two things, identify the differences. In compartmentalization with prokaryotes, which there isn't compartmentalization, and eukaryotes, in which case there is compartmentalization. And you can make an evolutionary connection to this. So this is where you're watching those videos that the College Board put out and you're saying, why am I watching about evolution if they cut Unit 7 out? Because it is interwoven to many of the big ideas, right? So right here we're talking about cellular components, and you have to make connections to evolution at this point. So we know endosymbiotic theory explains it explains compartmentalization, the enfolding of the membrane, creating endoplasmic reticulum. It explains mitochondria, chloroplasts. What's the evidence? So you can refer to DNA as an evidence, right? So the DNA of the mitochondria and the chloroplast is unique compared to the rest of the cell. It replicates on its own. The mitochondria and chloroplast both can replicate through binary fission, which is like a prokaryote, not a eukaryote. And it has similar, like the circular DNA rather than linear DNA. You could also talk about ribosomal structure, things like that. You should be able to compare eukaryotes and prokaryotes. When you're talking compartmentalization, you know that eukaryotes, they compartmentalize and therefore they're more efficient. Different areas of the cell can do different work. Prokaryotes kind of have a compartmentalization, they just don't separate it with membranes, so certain areas of the cell do certain jobs. When doing calculations, here's calculations that could come up in this unit. And again, I'm not sure how much calculation they would include in a 45-minute FRQ. So water potential, solute potential, just go back maybe and review those. You have a formula sheet. It tells you what each of the letters refers to. All you have to know is how to plug in the numbers, right? So just be familiar with that. Same thing with surface area to volume ratio. I'm not expecting that you would have to calculate that this year, but we do know that the larger the surface area, the better. So it's possible that you'd have to compare two cells. I don't know. Just, again, be familiar with your formula sheet. It is available to you. You can have that out while you're taking the test. So all the calculations are there for you. You just have to plug in the numbers. A couple skills that might be used in this is predict the cause or effect of a change in or disruption to one or more components in a biological system. So the cell is a system, right? Maintaining osmoregulation, that's a system. Generation of ATP, capturing of energy, all of those are systems, right? The electron transport chain is a system. So identify changes to any of those, disruptions with any of those, based on a visual representation or a biological concept, process, or model. And I think you can apply this skill to almost every unit. If the system changed, how would it affect the outcome, basically? Also be able to utilize data to evaluate a hypothesis or prediction, including supporting or refuting the alternative hypothesis. So with this skill, I think chi-square and standard deviation are your two examples. Again, be familiar with the formulas and how to use them. You have the formula sheet, so all you have to do is plug in the numbers. But if they're looking for statistical evidence, these are the two you use for statistical evidence. Okay, I think, power words. So knowing the difference between explaining and describing. So in a lot of those goals, it said be able to explain this or describe that. So here's the electron transport system, right? So this is, well, actually this is the light reactions, and then down here we have electron transport, but or chemiosmosis. Pick any of those. Try to explain it. by explaining it, you're making it understandable, the whole process. You're explaining how and why. You're going to include words like concentration gradient and diffusion or active transport. You're going to go step by step through a process and make it understandable to somebody else. Describing it is to make a mental picture of this so that somebody else can see what you're talking about in their mind. So again, when I'm done talking, just take this picture here and take a minute to explain it to somebody in your house. They're bored, they want to hear about it too. Or describe it. Or make a video and explain it or describe it to somebody in our class. But just, you need to practice, right? So when you see explain, know what they're asking of you, because if you describe instead of explain, you're not going to get your points. And also describe just is maybe a little bit simpler than explaining. Okay, so I think that's all I have for you on this unit. I hope that was helpful. I know I talk really fast, but you'd be bored if I went on any longer, right? So I hope you guys are doing well. I hope you're staying safe, making good decisions, make some chocolate chip cookies later, whatever you need to do, get some exercise, stay 6 feet away. Okay, see you guys all later.